Now let us know something about the isolation of bacteria or isolation of pure culture from a mixed population. In the nature, we do have microorganisms in a mixed population. For example, take any water sample or soil sample or milk, etc. Now these samples contains different types of microorganisms, and from these samples we can isolate pure cultures. Now pure culture is the one which contains only a single species, a bacteria which contains only of one species. So this is called as pure culture. Now coming to the various types of isolation methods, we will come to know now. Now to get a pure culture out of a sample, first we have to go for selection methods. So selection methods can be done by using physical method of selection or chemical method of selection or biological method of selection. In the case of physical method of selection, we can use different physical parameters like heat, pH or motility of the bacteria. For example, if you are considering a heat, you can take the sample. The sample may be a natural sample like soil sample. It is heated somewhere 80 degrees centigrade for half an hour. So most of the vegetative bacteria present in the soil sample are destroyed at 80 degrees centigrade, but the resistant forms, something like spore forming organisms like bacillus, clostridium, etc., they survive for this treatment. 80 degrees centigrade for half an hour is not going to destroy these spores. So that is how the selection, natural selection occurs within the sample. Okay, now let us know the effect of pH. Now some bacteria grow best at acidic pH, some bacteria grow best at normal pH, and some bacteria grow best at higher pH. So we can set a suitable pH based on our requirement, either low or high pH, so that the other bacteria are not grown in that media. That is how we can go for the selection of the required type of bacteria. Now similarly, motility can be used as one of the physical parameter for selection. Like you can just inoculate a media, semi-solid media on one side of that media with a mixture of the bacteria. The bacteria which are containing the flagella and which are motile, they swim across the semi-solid media and appear on the other side of the media. So this kind of separation occurs by using this motility as one of the parameter. Now we can use the chemical method of selection for screening the microorganisms from a mixed population of bacteria. Like we can use some inhibitory chemicals within the media, so that this is going to inhibit the most of the bacteria, and the bacteria which are resistant towards these chemicals are grown in that media. Similarly, biological selection can also be done, where some bacteria can be grown in some of the animals and some of the organs, and the other bacteria may not be surviving in that animals or in that organs. So by any one of this method, we can just go for the selection. By selection, we can reduce the number of bacteria from the natural samples. Like for example, natural sample may contain thousands of bacteria, but after selection, after employing the selection methods, the number will just come down to some few numbers, maybe 10 or even up to 100. Now from this, after selection, then we can go for isolation methods, and we can use any one of this method. It may be a streak plate method or pour plate method or spread plate method, or you can use micro manipulation method for isolation of bacteria into the purest culture. So we'll discuss one by one: streak plate method, pour plate method, spread plate method, and micro manipulation method.